It says, eventually, Billy came to dread his father's lectures over all other forms of punishment. There he is up there with the overhead projector, going through the transgressions. As I said, OH, alcohol group. And very typically, these are written, even when you're writing out molecules, you don't show the bonds within the groups in a lot of cases. It all kind of depends on how the group works. Everything potentially has changed. This is a fairly old overhead. You'll notice OH is called a hydroxyl group. It can be called a hydroxyl group. And so even if you know the names, sometimes you got to look up what somebody's talking about. Because depending on when they were trained or where they were trained, is that they may call something by a different name. Chemistry's been trying to standardize stuff for a long time, but I don't know, doesn't seem to have been terrifically successful, at least not within biology circles. I don't read a lot of pure chemistry papers, so maybe they're much better. They tend to be much more structure-oriented than biologists, so who knows? A double bonded oxygen forms two different types of groups depending on where the carbon is that it's attached to. If it's attached at the end, it's an aldehyde. If it's attached in the middle, it's a ketone. Let's see. That's showing a ketone. An acetyl group is a ketone just off of the end of the molecule where there's just a carbon and hydrogens on that end. That's a methyl group, right? Methane. And this turns out to be a very important functional group in protein processing, where the acetyl groups are, I'm sorry, I've got it backwards. Actually, it isn't important for protein processing. It also seems to, when you're trying to make a protein from the DNA sequence, it's a marker for the beginning of where you start pulling the code. So acetylation is a very important function in genetics, in genetic expression. The same thing's true of methyl groups. This can be used to activate genes, added or taken off of genes. I probably should have put those the other way around. Epigenetics. Epigenetics is changes in things, changes in what appears to be genetics that can be passed on. But when you look, what you find is a methyl group has been clipped onto the DNA. It's removable. The DNA itself is not changed, but the expression of that particular gene is affected by whether it's been clipped or not. So epigenetics is more genetic expression, but there are situations where the methyl group is clipped onto newly made DNA for offspring. It's sometimes called imprinting, where you send on the DNA with a gene already deactivated, can't use it. Carboxyl group, also sometimes called an acid group because that H doesn't like to stay on there. It comes off as a hydrogen ion, which of course acidifies the solution that it's in. So amino acids have this on the end. Nucleic acids have this on the end. All sorts of organic acids have this on the end. Speaking of amino acids, when there's a nitrogen group tagged on the end of the molecule, it's an amine or an amino group. Those seem to be used interchangeably. I'm trying to squeeze them in here. Crap. A sulfur group used to be called a mercaptan. Sometimes you see here sulfhydryl, and thiol seems to be mostly what you hear when you've got a sulfur group tagged on the end. A phosphorus with varying amounts of oxygen attached to it is a phosphate group. Adenosine triphosphate is got a string of phosphate groups on it. And then there's glycans. In complex systems, there's often strings of sugar molecules, varied lengths, sometimes varied sugars, that are called glycans. And those are very often used as detection sites, where how does your body know what molecules belong to you? Well, you've got proteins and stuff embedded in your membranes, but what really tells the difference between yours and maybe something from another source is a lot of the recognition that goes on is looking for the glycan strings attached to those marker proteins. I think that's, it's a glycan difference between the different ABO blood groups, for instance. Okay, that's the list of things. I don't know how much it's going to come up through our semester, but when you go on into organic chemistry, and if you're a biology major, get used to being a chemistry person also. My biology major had chemistry as what was called a second field. There was so much chemistry that it counted as more than a minor. It was biology with chemistry as a second field. That's very, very common. We have to take organic chemistry and biochemistry and molecular biology and lots of stuff that uses these wordings. So it's a good thing to have in your head. So you understand that.